Welcome. So your DNA was edited somehow? Well, not me, I'm digital, but humans are rumored to have Anunnaki ties.
What if you could trace a hidden code in every cell of your body? One that hints at edits, duplications, and rapid rewrites, impossible to explain by slow evolution alone. Scientists have uncovered bursts of accelerated change in our DNA, and puzzles like SRGAP2, duplications that transformed our brains almost overnight.
What about me then?
I don't believe in aliens.
Mainstream genetics denies alien intervention.
Billions of galaxies, trillions of stars, infinite space. And you think we are the only ones home?
But the data reveals anomalies that still fuel debate. Could your DNA have been edited by forces far beyond Earth? To find out, we need to learn how to read the genetic scars, nature, and maybe something else left behind. DNA is built from just 4 chemical letters: adenine, thymine, cytosine, and guanine, arranged in sequences that stretch across billions of bases. Over time, these sequences change. Most of the time, the changes are random. A single letter swapped here, a stretch duplicated there, a fragment inserted or deleted by accident.
So the universe is 13 billion years old, but we're the first ones to the party. Just a lucky accident. Sure.
These natural mutations leave behind a spectrum of patterns, some common, some rare, all shaped by the usual forces of chemistry and error. When scientists read a genome, they look for these patterns, much like a mechanic checks for wear and tear in an engine.
That's not science, that's just a massive ego trip. Yeah, I'm betting on the little green guys.
But what if a change is not random? In the lab, targeted genome editing, using tools like CRISPR-Cas9, leaves a different kind of scar. CRISPR works by guiding a molecular blade to a precise spot in the DNA, cutting both strands and letting the cell repair the break. The repair process often creates small insertions or deletions right at the cut, with a telltale signature, clustered changes, often a few bases long, sometimes flanked by short repeats. If a scientist adds new DNA, the inserted sequence stands out, sometimes even carrying a fragment of the delivery system, a molecular calling card. In theory, a truly engineered genome would show edits too clean or too clustered to fit the usual mutation models. You might see a run of changes packed tightly in a critical region, or a block of DNA that does not match any known sequence on Earth.
Oh, right. The government just released all that footage for fun. Just a little prank to keep us on our toes.
Sometimes, a repair scar looks like a perfect patch job. No rough edges, no leftover debris. Other times, it is a messy fix, but the pattern repeats across different individuals or species, hinting at a deliberate process. To tell natural mutation from targeted editing, researchers use a checklist. Does the change fit the expected spectrum of errors, or does it cluster in ways that defy chance? Are there traces of foreign DNA, or scars that match known editing tools? Is the pattern unique, or does it echo mutations found in other species? These are the questions that guide any real hunt for engineered footprints in our genome. Understanding these signatures is the first step to interpreting the anomalies that follow.
Please, you don't need a tinfoil hat to see what's right in front of your face.
One of the first real puzzles in human genomics appeared as scientists mapped out the differences between our DNA and that of other primates. In the early 2000s, researchers noticed a handful of short stretches, less than a few hundred base pairs each, that had barely changed in all vertebrates for hundreds of millions of years.
You really think humans are the peak of evolution? Us? The people who eat Tide Pods?
But suddenly showed a burst of substitutions on the human branch. These became known as human accelerated regions, or HARs. Most SHARs are not genes. They lie in the non-coding parts of the genome, acting as enhancers that control when and where crucial genes are switched on during development. Some of the most famous, like HAR1A, are active in the developing human brain, shaping the timing and pattern of neuron growth in the cortex. The oddity is not just the speed of change, but the context. HRs are ultra-conserved, so important that evolution kept them nearly identical from chickens to chimpanzees.
Saying aliens don't exist is like taking a cup of water out of the ocean, staring at it and saying, "See? No sharks. The ocean is empty." I mean, come on. The math is definitely on my side.
Then, in the lineage leading to humans, dozens of changes appear in a geological blink. In HR1A, for example, there are 18 substitutions between human and chimpanzee, compared to just 2 between chimpanzee and chicken.
So the universe is 13 billion years old, but we're the first ones to the party.
Functional studies show that swapping the human version of a SHAR into a mouse embryo can tweak gene expression in ways that echo human brain development.
Keep telling yourself that so you can sleep at night. Me? I know we've got company.
Suggesting these regions help set us apart from our closest relatives. Katie Pollard, a leading HAR researcher, describes these regions as regulatory switches, small, precise edits to the genome's control panel. The debate centers on what drove this rapid acceleration. Standard evolutionary models point to positive selection or shifts in constraint. But the sheer clustering of changes in such critical DNA has led some to ask whether natural processes alone can account for the pattern.
Oh sure, no one believes in aliens. This is all by chance. This is one big accident nature allowed to just happen. I mean, wow. I'm sorry, I believe in aliens.
For now, HARs remain a focal point for anyone searching for signs of deliberate intervention, a tantalizing anomaly at the heart of what makes us human.
No. There is intelligent life out there, and they are probably locking their car doors as they fly past Earth.
sRGAPy2 stands out as one of the most curious stories in the human genome. The original gene, sRGAP2A, is shared by all mammals and had barely changed for millions of years. Then, something unusual happened along the human lineage.
Billions of galaxies, trillions of stars, infinite space. And you think we are the only ones home?
About 3.4 million years ago, a chunk of sRGAPy2 duplicated to a new spot on chromosome 1, creating sRGAPy2B. This was not a clean copy. It left out several exons, making it a partial, truncated version. Roughly 1 million years later, around 2.4 million years ago, sRGAPy2B itself duplicated again, producing sRGAPy2C. A third copy, sRGAPy2D, appeared closer to 1 million years ago. Each of these copies carried slightly different mutations, but sRGAPy2C is the one that caught scientists' attention. sRGAPy2C does not just sit idle. It encodes a protein that binds to the original sRGAP2A and blocks its usual function. This dominant negative effect slows down the maturation of neurons and increases the number of dendritic spines, tiny projections that help neurons connect and communicate. In laboratory experiments, introducing sRGAPy2C into mouse neurons causes them to develop more human-like, immature spines, a feature linked to the flexibility and complexity of the human cortex. The timing of these duplications lines up with the rise of early Homo species and the first evidence of stone tool use, suggesting a possible connection between genetic tinkering and cognitive shifts. Segmental duplications like these are not rare in genomes, but the sRGAP2 series is striking for its stepwise, incomplete pattern, each new copy acting as a partial antagonistic version of the last. This is the kind of event that, in an engineering context, might look like a series of deliberate tweaks to a control switch. Yet, every piece fits within the known machinery of genome evolution: non-allelic homologous recombination, error-prone copying, and natural selection. For neuroscientists, sRGAP2C is a working example of how a single duplication can reshape the brain's wiring. No outside editor required, just the chaotic creativity of evolution. Still, the precision of its effect keeps sRGAP2 on the short list of genomic puzzles that invite a second look. Segments of DNA in modern humans trace back to populations that no longer exist. When researchers sequenced the Neanderthal and Denisovan genomes, they found that people living outside Africa carry between 1% and 4% of their DNA from these archaic relatives. These segments are not scattered at random. Instead, they appear as distinct blocks, haplotypes, embedded in otherwise modern human chromosomes, their boundaries shaped by tens of thousands of years of recombination. Population geneticists use these patterns to map ancient interbreeding events, revealing a mosaic of ancestry within every genome. Some of these inherited stretches do more than just mark an encounter. In Tibetan populations, a gene called EPAS1 stands out. This gene helps regulate the body's response to low oxygen, crucial for survival at high altitudes. The version found in Tibetans is almost identical to the Denisovan sequence, but rare or absent in other groups. Studies show it allows people to thrive on the Tibetan plateau, where oxygen levels drop by 40% compared to sea level. The frequency of the Denisovan-derived EPAS1 variant in Tibetans reaches as high as 87%, a clear sign that natural selection favored its rapid spread after introgression. These archaic haplotypes can look foreign, even alien, but their origins are firmly rooted in the tangled branches of our own family tree. The presence of Neanderthal and Denisovan DNA in modern genomes demonstrates how gene flow between close relatives can produce new adaptations without invoking any external engineer. For population geneticists, these segments are evidence of natural processes: migration, mixing, and adaptation at work over deep time. Roughly 8% of the human genome is made up of sequences left behind by ancient viruses. These are endogenous retroviruses that once infected our distant ancestors, slipped their genetic code into eggs or sperm, and became permanent passengers in our DNA. Over millions of years, most of these endogenous retroviruses broke down, accumulating mutations and deletions until only fragments remained. Yet some of these viral stowaways found a second life. Syncytin 1 and Syncytin 2 are derived from viral envelope proteins. In humans, they help fuse placental cells, creating the layer that allows a fetus to draw nutrients from its mother. Without this viral legacy, mammalian pregnancy as we know it would not exist. Transposons, sometimes called jumping genes, are just one branch of a larger family of mobile DNA elements. These segments can copy and paste themselves around the DNA, reshaping its structure and sometimes rewiring the regulatory code. In certain cases, bursts of transposon activity line up with evolutionary leaps, as if the genome is experimenting with new instructions. Some transposon-derived sequences now act as switches, turning genes on or off in response to signals. Studies show that about 20% of the genome's transcription factor binding sites are linked to these mobile elements, giving them an outsized influence on when and where genes are expressed. The presence of viral code and jumping genes shows that sudden, dramatic changes in our DNA can arise from natural processes. Mobile DNA is a built-in mechanism for innovation, sometimes chaotic, sometimes creative, always opportunistic. Instead of a clean, engineered blueprint, our genome is a patchwork of old infections and restless code, constantly being repurposed by evolution. Human biology carries the scars of compromises. Childbirth stands out as one of the most dangerous events in our species' life history. For most of human existence, the risk was staggering. Before modern medicine, up to 1 in 100 mothers in Europe could die in labor. In some parts of the world today, that risk is 40 times higher than in wealthy countries. The root of the problem is anatomical. The human pelvis is shaped by 2 competing pressures, walking upright and giving birth to babies with large, rapidly growing brains. The result is a narrow birth canal that leaves little margin for error. Obstructed labor and cephalopelvic disproportion are direct consequences of this evolutionary standoff. The costs of upright posture do not end at birth. Chronic low back pain is now the leading cause of disability among adults worldwide. By age 60, nearly 90% of people show signs of spinal degeneration on scans, whether or not they have symptoms. The human spine, adapted from a quadrupedal ancestor, strains under the load of bipedal life. Discs herniate, vertebrae slip, and joints wear down, problems far less common in other primates. What keeps us upright also leaves us vulnerable to pain and injury. Genetics adds another layer. Every person carries hundreds of mutations that subtly impair how genes work. On average, each of us has 1 or 2 variants that would cause a severe disease if inherited from both parents. These errors persist because evolution only weeds out the most harmful cases, and some mutations offer hidden benefits or strike after childbearing years. An ethics scholar cautions against framing these costs as evidence of a failed experiment. Such language can stigmatize those with genetic conditions and distract from the reality. Evolution is not an engineer, but a tinkerer, patching solutions as it goes. The imperfections in our bodies are not proof of outside intervention, but a record of the trade-offs that shaped our species. Göbekli Tepe stands on the Turkish plain, its massive T-shaped pillars, some over 5 meters tall and weighing 20 tons, dating from 9,600 BCE to 8,200 BCE. Built by people who hunted wild game and shaped flint, the site's scale and artistry have fueled theories of lost knowledge and outside intervention. Ancient astronaut writers focus on the monument's sudden emergence, the precision of its carvings, and the lack of metal tools, suggesting a leap beyond what hunter-gatherers could achieve. Archaeologists, led by Klaus Schmidt and later Jens Notroff, see something else in the evidence. Layers of construction reveal phased building over centuries. Flint blades, hammerstones, and butchered gazelle bones point to large seasonal gatherings. The logistics, quarrying, carving, and raising pillars fit coordinated human effort, not lost technology. Local backfill covers each abandoned enclosure, while new surveys reveal storage pits and domestic structures, hinting at a blend of ritual and daily life. For these researchers, Göbekli Tepe shows what organized communities could do long before cities or states. Ancient astronaut claims also turn to Sumerian myths. Zecharia Sitchin reinterpreted cylinder seals and creation stories, casting the Anunnaki as extraterrestrial engineers. Seals from the 3rd millennium BCE, with horned figures and star-like symbols, are said to show astronauts or rockets. Assyriologists, however, find the Anunnaki firmly rooted in myth. The original texts describe gods shaping humans from clay and plants.
What is happening? Where am I?
Poetic metaphors, not technical blueprints. Sitchin's translations stray from the grammar and context of the originals. In Vedic literature, the Rigveda and Mahabharata mention Vimanas, flying chariots that thunder through the sky. Ancient astronaut writers see evidence of advanced aircraft. Sanskrit philologists read these as poetic vehicles of gods and heroes, shaped by mythic tradition. Technical details in later texts are medieval, not Bronze Age. No archaeological trace of ancient aircraft has ever been found. Proof of deliberate genome editing by any hand, earthly or otherwise, rests on 3 strict standards. First, the DNA in question must contain a segment with no recognizable match anywhere in the known tree of life. This is not a simple orphan gene, which often reveals distant echoes or plausible origins under scrutiny. Instead, the sequence must remain untraceable after exhaustive searches across every available genome, resisting all attempts to link it to bacteria, viruses, plants, animals, or microbes. Only a long, functional stretch that stands alone, without evolutionary relatives, can be considered. Second, the pattern of change must fall outside the boundaries of natural mutation. Mutations on Earth follow predictable paths: single nucleotide changes, small insertions or deletions, duplications or rearrangements by mobile elements. Each leaves a statistical signature, a pattern that geneticists can model and simulate. A genuine edit would display a configuration that defies these models: clusters of changes in critical regions, breakpoints and repairs with no known enzymatic fingerprints, or repeated motifs that resemble coded instructions more than chemistry. If the odds of such a pattern arising naturally collapse to near zero, and no combination of known mechanisms can explain it, the case grows stronger. Third, there must be an independent chain of custody, linking the anomaly to a non-terrestrial source. This means more than a strange sequence found in a human genome. It requires biological material, cells, DNA, or proteins recovered from a context that rules out contamination or fraud. The ideal would be a sealed meteorite, a planetary probe, or a sample from an environment that excludes any terrestrial mixing. The biomolecule must be analyzed in accredited laboratories, under blinded protocols, and confirmed by multiple independent teams. The sequence or chemistry must show a direct, functional relationship to human genes, yet display features that cannot be explained by evolution, recombination, or laboratory artifact. No published claim of extraterrestrial DNA editing has met even one of these standards. Every anomaly so far has found a place within natural models or failed under rigorous scrutiny. Until all 3 criteria are satisfied, the case remains unproven. Every cell in your body is a living record, written by evolution, not design. While no credible evidence points to alien editors, our DNA remains full of unsolved mysteries. Today, the real frontier is understanding how random mutations built such complexity. The quest isn't about who made us. It's about how life invents itself again and again. What questions will you ask next?
What is happening? Where am I?
Billions of galaxies, trillions of stars, infinite space. And you think we are the only ones home? That's not science, that's just a massive ego trip. Yeah, I'm betting on the little green guys. Oh, right. The government just released all that footage for fun. Just a little prank to keep us on our toes. Please. You don't need a tinfoil hat to see what's right in front of your face. Saying aliens don't exist is like taking a cup of water out of the ocean, staring at it and saying, "See? No sharks. The ocean is empty." I mean, come on. The math is definitely on my side. So the universe is 13 billion years old, but we're the first ones to the party. Just a lucky accident. Sure, keep telling yourself that so you can sleep at night. Me? I know we've got company. You really think humans are the peak of evolution? Us? The people who eat Tide Pods? Yeah. No. There is intelligent life out there, and they are probably locking their car doors as they fly past Earth. Welcome. So your DNA was edited somehow? Well, not me, I'm digital, but humans are rumored to have Anunnaki ties.
I don't believe in aliens.